Good afternoon and good evening. We get into our lecture, one more in the series. I find there are more select people in the audience than before. The title of the talk is shown to you in the first slide. Am I audible or do you want me to be louder? The third alphabets of life. The second part, maybe we may discuss, we may not discuss synthetic vaccines as opposed to biological vaccines. I had taught this subject to postgraduate students in the Regional Center for Biotechnology. Before that, I had read a very large book by an Indian author from abroad, Verkey's book, and I had seen a video many times to which I will make a reference in this lecture. A very wonderful video. I saw it many, many times. So as we go along into the lecture, what are the third alphabets of life? Are we not curious? But if we have to talk about the third alphabets of life, that it presupposes that we know what are the first two alphabets. Now, there may be a debate on the choice in the next slide where you are shown some points for this lecture, that the other two alphabets which precede this third alphabet are very well known to us. Right? Yes, some of the points that will be discussed will be shown to you shortly. We will naturally ask, what are the third alphabets of life? We will ask, what are sugar codes? And how are they possibly going to run computers for the future? I don't know. There is this swine flu, which is connected with a very interesting acid called stearic acid. A new word will be introduced to you, glycans. And the last part refers to synthetic vaccine. What are these? And can there be a fuller azir or fuller azir vir? You know, whenever you put vir, it means it is an antiviral. And sugar balls, nano sugar balls. These are some of the things that you will hear. Maybe the last two titles we may not get into very much, may not have the time. Yes, there are these first two alphabets shown. What is the first one on the left-hand side? Now, A and B can be exchanged with each other depending on your line of thinking. If you like nucleotides, which go the bases which go to make DNA, RNA, and so on, then that is shown in the first slide. That is the nucleotides. But the problem is nucleotides can undergo extension only in two directions. 3'N, 5'N. 5'N is the CH2OH, which is shown. Chain elongation can take place from these two directions, presuming that what is shown is a 2-deoxyribose. There is no oxygen in second position. The molecules shown in part B of the slide refer to alpha L amino acids, which go to make the human body, the life that we know on this planet. I am having fundamental doubts why we are restricting to only 20 or 21 amino acids. Are we that select? But notice, chain elongation in amino acids can happen via the amino group on the left-hand side, the NH2 group, or the COOH group. So chain elongation can happen largely from only these 2 points. On the part C is shown a chair conformation of a pyranose sugar. And you see multiple points for chain elongation. Why is chain elongation so important? DNA, can it be called a polymer? Can peptides and proteins be called polymers or macromolecules? Same way, when carbohydrates like the one shown in C 1, 2, 3, 4, 5 different arrows are shown marked, or 6 arrows are shown. That means in 6 directions, chain elongation can take place. I don't want to say necessarily polymerization. I use the phrase chain elongation. So conceptually, this is very important, that we are going to focus on part C. And even in the postgraduate class where I took this course and taught them, I found many students who had done biochemistry, biotechnology, were not familiar with it. Is biochemical coding like a language? Can you write words and sentences and the order of letters, whatever those letters are, can it be used to store information? Is there high density coding possible? Is it something like the postal code? I do not know how many in the audience know what is the PIN code of St. Stephen's College, Delhi 110007. I do not know, many of you come from different parts of Delhi or India, and you may have your own postal code. What is the importance of postal code? Even a machine can sort out the letters and send it to the right place. Therefore, this particular topic that we are discussing today is discussed under sugar codes. That means the way we are going to write the names, we'll use those either alphabets or names or some figures like circles, rectangles, squares. The next slide, you will see some of these coming up. That is, we can write sentences using these sugar codes. We are emphasizing on the word sugar, but it's incorrect in this discussion because you're going to see very soon that it is not only sugars that are involved. Yes? See, we are talking about a substance or compounds which we do not read in MSc Chemistry, BSc Honours Chemistry, but some biochemistry students are familiar with it. A glycan, we are talking about glycans. Believe me, glycans are referred to as the third alphabet of life. This can be read by a lectin, and this can be used just like a postal code by a courier service or a delivery service. There is another word, phyto, plant agglutinin, synonym for plant lectin. Lectin reads the sugar code. Is there a mammalian lectin? Yes, from rabbit liver, they have isolated the first mammalian lectin. Lectins read these codes. So we are not yet defined a glycan. We should do so very quickly. And that is, as we see the next slide, you can move on to the next slide. You see, these contain many sugars. There is one more part of glycans which I have not highlighted yet. It will come in one of the next slides. Glucose in this sugar code is shown as a circle which is fully filled with a color. I do not know what is the color that you see. Galactose is again a circle, but with a different color, yellow color. Mannose too is a circle, but color is different. The three which have circles with different colors represent glucose, galactose, mannose. Chemistry students are familiar with these three names. But chemistry students are not familiar with sialic acid, which is shown as a red diamond. We don't write sialic acid. We don't write its structure. For the purposes of studies, we can show these circles, diamonds, fully filled with a color or half filled, as is the case of glucuronic acid. Another carbohydrate chemistry students are not familiar with is fructose. I think there is a C missing. Fructose. Below that, you see that's a triangle with a color, possibly red, if you can see it. And then there is N-acetyl glucuronic acid, or the N-acetyl galactose, or xylose. These are some of the sugars which are important in the studies on glycans. But we have to define the word glycan yet. I don't know whether it is shown in the next slide, but you may see it. Let me anticipate it as the slide is made to transit to the next one. The definition of a glycan is carbohydrates like these ones bound to proteins. But the definition keeps nucleotides out. The International Union, which has taken the decision to define glycans, the next slide, I hope, as these carbohydrates bound to proteins. This is sialic acid. This is involved in the receptor for influenza virus. And this was described as early as 1966. In the chemistry class, you know, glucose, galactose, fructose, sucrose, lactose, maltose, sucrose, starch, but we do not know sialic acid. If you notice, the number of carbon atoms is 5 carbons in the ring, 1 carbon outside in position 1, 6, and a carbon here too on the 1, 2, 3, 4, 5th position. There are 5 plus 1, 6, plus 3 carbons. That makes it a 9 carbon sugar. We do not discuss such stereospecific molecules like sialic acid in a chemistry class. But it had already become very important pre-COVID days. But during COVID, the importance of this became many, many fold. And sialic acid, I repeat, is a 9 carbon sugar. There is an amino group which is acetylated. There is a carboxyl group. Like always, there are hydroxyls, and the hydroxyls are positioned in a very specific manner, either axially oriented or equatorially oriented. I know this is not part of BSc honors chemistry or MSc chemistry, but biochemistry students do get familiar with these things, but not in a big way, because the level of education in many biochemistry, biotechnology classes, as far as stereochemistry is concerned, is usually a low degree of knowledge, whereas students of chemistry are very strong. What a beautiful structure, very complicated structure. This is called an N-glycan core. But this structure is incomplete because this must be attached to a protein. And there is a core pentasaccharide, 5 glucose carbohydrates or sugars in black color. There are others in blue color. It looks like antennae attracting something or going to attach to something. You will shortly understand why it is so important. But there is a protein attached, and often the protein is attached through amino acids of the protein like arginine, which is a basic amino acid. This is a complicated structure. 1, 2, 3, 4, 5, 6, 7, 8, 9 sugars. Very complicated with very precise stereochemistry. It appears on the left-hand side, there is something like an insect wanting to bite you and grab you. Yes, this is an example of an N-glycan, because on the right-hand side, the bonding to the protein is through the N terminal of the protein. Yes. Next slide is shown something that we have never heard of. All of us talk about soya bean. This is an agglutinin called soya bean agglutinin. Its structure is shown on top, and it is an N-acetyl glucose, 2 units with so many units, 9 units of mannose. I don't know, with a very specific branching, alpha 1, 2, alpha 1, 2, alpha 1, 6. We know this in disaccharide, that means in sucrose, how fructose and glucose are bound in sucrose, how the glucoses are bound in, maltose, how the glucose and galactose are bound in lactose. This all we study within our BSc honors course. But a molecule like this looks terribly complicated. Next slide shows you. Actually, influenza virus, this photo you see, which is called as H1N1 virus. You'll see what H stands for. You'll soon see what N stands for. It's also called swine flu. The influenza virus requires its attachment to sialic acids on cells. When the cell is being attacked by a virus, this virus needs on the outer surface of the cell presence of sialic acid. I do not know. I have not tasted sialic acid. I do not know whether the pronunciation sialic acid is also correct. You can correct me if you think there is a better pronunciation. But you saw its structure with 9 carbon atoms. ऐसा लगता है कि वायरस को ये मीठा लगता है, वायरस को ये बहुत पसंद है. The virus comes and binds to sialic acid. That is the early step for influenza or other viral diseases to happen. Many things happen subsequently. That means the virus has come and it is attached to the outside surface of the cell, and the point of its attachment is on a sialic acid unit. Then it starts doing some more things. It binds to another part which is called heme-agglutinin. You will see that, and this is beautifully explained by Caroline Bertozzi. I wrote this article in Current Science from Bangalore, published by Indian Academy of Science, Bangalore in 2019. It was published after very careful corrections and editing by the referee and by the editors. Current Science 2019, you will see my article, The Third Alphabets of Life. And you will see that there is a reference to a video by this lady by name Caroline Bertozzi. At that time, when I wrote to her, she was not yet a Nobel laureate. This has happened with me many times. When I finished my MSc, I was in touch with Professor Adolf Butenandt, who was actually a Nobel laureate at that time. I went to Irwin A. Rose in Philadelphia. After I met him, he became a Nobel laureate. I didn't work with him. I worked with a student of Adolf Butenandt, who won the Nobel Prize for his work on steroid hormones. I went to, just like Irwin Rose, I went to another person who came to St. Stephen's College. I told you his name was the man who has made molecular switches. And you see his name also, I mentioned the other day, starts with B. Ben Feringa. Ben Feringa, he made molecular switches. How do you locate people before they win the Nobel Prize? Before they become famous? In your days, it's not so difficult as in our days, because we did not have mobiles, we did not have computers or laptops, we didn't have access to Google or any other search engine. We had to go to the library, sit down, with chemical abstracts, search for paper by paper, and send them requests to send their reprints. Some of them did agree. The next slide, we see that this particular virus, what is it going to do on the cell surface? I told you, this is a short screenshot from Caroline Bertozzi's lecture, that it is going to sit on the surface of the cell. The influenza virus is binding. After some time, it says, I forgot. I have to bind with hemagglutinin. Oh, I will now bind with hemagglutinin. Then it says, but I have to get out. I have to get into the cell, because then I have to take over the cellular machinery, capture its DNA, start making my proteins, and infect the organism where I came and attached myself. Oh, but I have not cut off my original bond to sialic acid. Oh, I forgot to cut it. So that at a later stage, that sialic acid bond is broken. And the virus moves into the cell and takes over the cellular machinery. Because there is a hemagglutinin involved, H1, there is a neuraminidase which actually cleaves the bond of sialic acid with the virus. So this is what is called H1N1 virus, also known, I presume, as, and what is it? You call it swine flu or influenza virus. Some of us have read about how viruses invade. You see that sialic acid molecule is shown in the insect. How is it going in and forming a bond? Yes, this is taken from Caroline Bertozzi's video, is short, and we have seen the video in a classroom situation. But today, I am not going to show the whole video. If at all, we will try and show you a part of the video. But you may go home and look for the link and see the entire video a few times for you to understand it. Yes, in the next slide is shown.
Okay. So here's a cartoon that illustrates the anatomy of the influenza virus.
That is Caroline Bertozzi, a very smart lady. At the time I was showing this in my classroom, she was not a Nobel laureate. But in 2022, along with Sharpless and Meldal, she won the Nobel Prize because she did something which the other two did not do. She attached a fluorescent label to her molecules which are undergoing Bertozzi reaction. I'm not saying click reaction in the strict sense of the term, but bio-orthogonal chemistry inside living cells. So go ahead and listen to her.
It's a membrane-enclosed virus that has a core that has both RNA and proteins. But there are two proteins that sit on the membrane envelope of the virus. And those proteins go by the name hemagglutinin, which I abbreviate H, and neuraminidase, which I abbreviate N. And remember, the swine flu is more scientifically termed H1N1. Well, the H1 is a certain form of hemagglutinin, and the N1 is a certain form of neuraminidase. Now, we know quite a bit about what these two proteins do. In fact, we even know their molecular structures in very great detail. Hemagglutinin is a receptor. It's a protein that binds to a sugar, and that sugar happens to be sialic acid, which I mentioned before. Neuraminidase is an enzyme. And what neuraminidase does is it catalyzes the cleavage of sialic acid off of the host cell. So this protein attaches to sialic acid, and this protein cuts the sialic acid off and throws it away. Now, when that discovery was made, it struck many scientists as a paradox. Why would the virus have a protein that attaches to sialic acid and yet another protein that just cuts off that sialic acid and tosses it away? Well, I'll show you what those two proteins do. It turns out that hemagglutinin is important in the very first stage of infection, where the virus lands on a cell. It's a human host cell. The hemagglutinin attaches to the sialic acid and basically allows the protein, or I should say the virus particle, to dock on the cell surface. Once that occurs, it triggers an endocytosis event, where the host cell inadvertently engulfs the viral particle into a vesicle. The membrane of the virus fuses with the membrane of the vesicle and releases the nucleic acid into the cell. And now that viral nucleic acid takes over the machinery of the cell and forces the cell to generate more viral particles. Those viral particles assemble around the membrane of the host cell, and eventually a new viral particle buds off the cell surface, as I showed in that previous electron micrograph. But remember that with all that hemagglutinin around, that viral particle might get stuck on the cell surface where the sialic acids are. And so the job of neuraminidase is to cut those sialic acids off at that point so that the virus can release itself from the cell and go find another host cell to infect and complete the cycle. So that's why we need these two proteins that act on sialic acid. Now, knowing the importance of neuraminidase in the viral life cycle, many scientists thought that if one inhibits that enzyme and prevents this very last step in the cycle, one might be able to shut down the propagation of the influenza virus. And so a large drug discovery effort was underway back in the 1990s, even the late 1980s, to develop inhibitors of the neuraminidase enzyme. And that was done by understanding the mechanism of that enzymatic reaction. So the mechanism is shown here. Here's a sialic acid, and picture it bound to the surface of a cell through a glycan on a glycoprotein or a glycolipid. So the R group is the rest of the glycan or the rest of the glycoprotein. What happens during the neuraminidase catalyzed reaction is that there's a cleavage of the bond right here between the sugar ring carbon and this oxygen. That's called the glycosidic bond. And what the enzyme does is it finds a way to make this bond reactive, so this bond is cleaved, and there's a transition state for this reaction in which there's basically a change in the hybridization of the carbon atom at this position, so that it goes from being what we call sp3 hybridized to sp2 hybridized. It becomes planar, and also a positive charge develops on the ring. And then that leads to the formation of this intermediate, and then water from the environment reacts with the intermediate to form a free sialic acid molecule, which then floats away. Well, what several pharmaceutical companies did is to look at the structure of this presumed transition state and try and mimic that structure with these synthetic molecules that are somewhat reminiscent of sialic acid. For example, this compound has the sp2 hybridization at this carbon, similar to the transition state. And so does this compound. This compound has a positive charge in the form of this guanidino group. And this compound has a positive charge in the form of this amino group. These two molecules are actually now on the market as flu drugs. This compound goes by the trade name Relenza, and this compound goes by the name Tamiflu. So if you feel the very, very early symptoms of the flu coming on, you can go to the doctor, get prescription for one or the other of these, and try and prevent the full-blown onset of the flu. Or if someone in your family has been diagnosed with the flu and you're worried that you might catch it, once again, you might take one of these two drugs as a preventative measure, as a prophylactic against the flu. So this is a nice example where understanding the glycobiology of influenza led ultimately to the development of drugs to treat the flu. It's a very nice story.
The lady, Caroline Bertozzi, was not a Nobel laureate when I was studying her work in 2016, 2017. But she won the Nobel Prize in 2022. By that time, more than one batch of MSc and PhD students had taken these lectures from me and had to learn the structure of sialic acid. What are the two key elements? Key elements are the way the virus comes and attaches itself to a sialic acid unit. And that is on the outer side of the outer surface of the cell. And how, in stepwise fashion, the virus enters into the cell, takes over the machinery, and starts making its own copies and sends them as the infection inside the entire body. But at that stage, I repeat what she said, the bond with sialic acid has not yet been cut. So enzyme is needed for this. And the enzyme is mentioned here as NUR aminidase that cuts the sialic acid. Very crucial for the process of infection to continue, to spread. But the problem is that the intermediates in the transition state involved have more of an sp2 character. That means there is a generation of a double bond. So if we can mimic the transition state with some chemical molecule, it may serve as a drug to control the disease. This is exactly what happened. And you will see in one of the next slides. I'm sure you are impressed the manner in which Caroline Bertozzi was talking and given her talk in her own university. And also there are some other videos which you can refer to. So we go on to the next slide. And remember, we are going to look at one of the two or both the structures she referred to. Yes, this is containing a double bond, the 6-membered ring. This is the drug which is marketed as Tamiflu. The cyclohexene ring, substituents, 3 different substituents. It mimics the transition state involved in the cleavage where sialic acid is being broken away or taken away. Next slide. There is another drug shown here, and you can see that it has an oxocarbonium intermediate, that it has a positive charge, it has a double bond, yet a positive charge, and this drug works as a H1N1 or an influenza virus drug. So just by asking questions, how do viruses work? The IUPAC, IUB definition of glycans is protein bound to carbohydrates, other than nucleotides. Nucleotides are not taken into this discussion. We have not focused. We are focused only on the carbohydrate part. We are not focused on the protein part. Protein is a huge protein. You'll see some of them shortly, but beware. Some of the drawings and the figures could be dangerous. I show you the next slide. We spoke about this is harmless. You can make this agglutinin just like heme agglutinin, which was mentioned in the slide and in her lecture. Soya bean agglutinin can be prepared overnight. Its structure is shown in the middle. It is a good source of manganese, 9 manganese units, and an N-acetyl, I hope, glucose, amine or glucose, glucuronic acid. You have to check on the GLC. It is used in bone marrow transplants, and during the transplant, one purges with soybean agglutinin. This is done routinely in hospitals. So this is a glycan which is repeatedly used in hospitals. There are some children who are referred to as bubble children. A particular name of a child is mentioned, that is our David Vetter, and he actually was a child born with this deficiency in 1971, and he had, a very bad immune deficiency, and he had to be kept in what we call today something like a ventilator. We didn't know these words previously. So they have to be kept in a very protective environment. And you will be happy to know it is not only soybean agglutinin, which is a glycan, which is very important and very useful. It saves the lives of leukemic patients, but even our own pineapple stem. Now, I'm not referring to the pineapple fruit. Please understand me. I'm not referring to the pineapple fruit. I'm referring to the pineapple stem. Pineapple stem, which is very cheap and is usually thrown away, contains a very wonderful glycan known as bromelain. It has no bromine, but it gives you a glycan which can be very, very pure You can purify this. This is a glycoprotein. Glycoprotein means sugar plus protein. Glycoproteins are glycans. They are the third alphabets of life, to be clear. That means pineapple stem can be used. I'm not going to explain this. Many students in school read this blood grouping. And your blood group is very, very important. The donor must have exactly matching blood group. Otherwise, there is disaster. Here too, if you notice, the sugars that are mentioned here include, if you can read, there is a glucose, there is an N-acetyl glucosamine, there is a galactose, there is an N-acetyl galactosamine, and there is fructose. But one of the two groups differ only where you have a small change, and that makes a difference in the blood group. Knowing our blood group is very important, and the blood that is to be given or transfused or infused, given to some patient, the matching must be done perfectly, otherwise it leads to disaster. Next slide. So glycans are very important, even in matching blood that has to be given. You know, our St. Stephen's College, I have seen every year the Red Cross comes and collects a lot of blood from our students who generously donate, but that has to be preserved and also matched. NH-acetyl is present in a blood group B, but in the other one, you have an OH. In A, you have NHAC. NHAC means amine acetylated, and that becomes a hydroxy. This is the minor difference between one blood group and the other group. A small bit of chemistry there shown. I'm not explaining it in detail. As we go to the next slide. That's what I was saying. It's going to be dangerous. I don't know whether this is from India or from some other neighboring country. I'm sure there is nobody in this audience who has been bitten by a scorpion. If you go to the Indian Institute of Science, Bangalore, when I went there first time, 1973, to attend a 1-month summer course, the huge scorpions going around the place even today. But they seem to be shy. They don't come and bite you. But you know, it contains a glycan, which is known as hemocyanin, and the composition is shown on top. And a lot of such compounds have been described in scorpions from different places. So I'm not going to mention the other countries. So they are peculiar to each country and each place. I don't know why scorpions are so dangerous, but they contain a hemocyanin. I do not know whether the color of the blood of scorpion is just like that of the human being. I raise a question. Next slide. So I think we have covered our main topic today in less than 45 minutes. I may go into this and towards the end show a slide. But before we go back to the previous slide, so we do not move to the next part of our talk. To summarize, we have defined when you have carbohydrates like sialic acid, fructose, galactose, glucose, and others, about 9 of them or 10 of them or more, they are all nowadays represented by codes. These sugar codes can be read just like PIN codes, which we put for our courier. He wants a PIN code, the courier man, he wants a mobile. So similarly, these various glycans can be put up as sugar codes, which can be understood so that if a molecule is to be delivered in some place, it is delivered precisely. But it is not just carbohydrates. Carbohydrates bound to proteins, which have not been shown in this lecture so far. It's a large molecule, but these sugar codes which are the glycans, have antennae. I'm not talking about the antenna of the scorpion, which is where it bites, but I have seen people bitten by a scorpion. They suffer so much. So glycans are not only harmful in some cases like the scorpion bite, but also very useful like the soya bean agglutinin, or I dream that me and my students will work with bromelain from pineapple stem, and these could be used to treat viral diseases. Are you hinting, sir? You may ask, you are talking only of influenza virus. You are talking about H1N1 virus, swine flu. Are you hinting that some such things are or could be important in even treating a pandemic like the one that we had called as COVID-19? So I think we are ready to go into the part 2, which is not the main focus of our talk. There are some concepts known as synthetic vaccines. How are synthetic vaccines different from the vaccines that we heard so much about during the COVID pandemic? The difference is synthetic vaccines are made by chemists, and they consist of peptides and carbohydrates or antigens and are considered safer than those made in the biological cultures. Sometimes the vaccine that we have had, we all know that there were problems with some of the vaccines in the country and in other countries which have now come out in the open. But these chemically made synthetic vaccines will have precise structures, and you can produce them in a reproducible manner. Many of them have shapes which are like a Y-shaped immunoglobulin, T cells and so on, and the B cells which are part of our immune system. That means we need not talk of biologically produced vaccines which dominated the discussion, but we could talk of synthetic vaccine. I'm not the only one talking about it. Even President Trump, when he had not yet suffered from COVID-19, went to see the lab of a person who was producing synthetic vaccines in the United States in a university. I forget the name, maybe the name figures somewhere in my slide, and had a look, but he did not take a synthetic vaccine to treat himself. He took what I mentioned to you earlier as a immuno, what was that I talked to you the other day? That means I said monoclonal antibody. He got himself treated with a costly monoclonal antibody. Next slide shows us a synthetically made molecule via the click reaction, 2022 Nobel Prize, and it has an overall Y-shaped. You can make any shape in the laboratory, and they naturally resemble the immunoglobulin so important for our immune system, which is compromised in many of these diseases. We were confident that we could make such a molecule ourselves. That is the purpose for my introducing this photograph of a Y-shaped molecule. You will see shortly, there is a definition. Vaccines, Manurba, why don't you read this out nicely as you did the other day? Please read it.
Okay, sir. So, vaccines have typically been prepared using whole organisms. These are normally either attenuated bacteria or viruses that are live but have been altered to reduce their virulence, or pathogens that have been inactivated and effectively killed through exposure to heat or formaldehyde. However, using whole organisms to elicit an immune response introduces the potential for infections arising from a reversion to a virulent form in live pathogens, unproductive reactions to vaccine components or batch to batch variability, Synthetic vaccines in which a molecular antigen is conjugated to a protein offer the opportunity to circumvent these problems.
This paper was published in 2015 in Nature when nobody was talking about a pandemic. Next slide. But synthetic vaccines are available now, but they are not yet, strictly speaking, that popular. In Washington State University, Dr. King, on the 9th of March in 2020, in the midst of COVID-19, in Scientific American, reported that to develop a coronavirus vaccine, synthetic biologists tried to outdo nature, and that synthetic biologists, you can say synthetic chemists, can do better than nature. This asserts that synthetic vaccines made by binding peptides, carbohydrates, or antigens to obtain shapes resembling cells of our immune system could help control viral diseases, including COVID-19. But we were in such a great hurry, next slide, that in India, we were not prepared to consider synthetic vaccine as a possibility. In 2012, the European Journal, Chemical European Journal, was published a paper. There is a Sumati Bhatia in Chemical Communications in 2014. There is another paper by Taylor in 2017. At that time, these people were making it. There was no pandemic. They were trying to make what are called nano sugar balls. We also propose, can we make a nano sugar ball? This nano sugar ball that is shown, is there another slide? Go back. Yes, this is good. In the second lecture, yes, next slide. The second lecture, I spoke about fluorazirman. Fluorazirman is a manganese complex with 3 carboxylic acid units and 3 amino groups donating electrons for the formation of this complex. If we were to break the manganese away, to break manganese is not difficult. You can treat this complex with alkali, or you can pass H2S gas. I hope most students still do qualitative analysis in the chemistry lab in undergraduate days, so they all know the smell of H2S, how horrendous it is. Manganese sulfide gets precipitated. You get from fluorazirman a molecule without manganese. So fluorazir. Then what we did was, you go back to the previous slide. In our lab, we did this. We attached units of sialic acid via a process of click reaction, which was discussed previously and referred to today. So that 1, 2, 3 triazoles, 1, 4 disubstituted triazoles, all around the ring. If you count, I don't know how many, a huge number of these sialic acids are sitting on the outside of the fullerene molecule. This looks beautiful. That means it can actually capture a coronavirus. We call this molecule fluorazir-bir. We call it fluorazir-bir. It's no longer fluorazirman. We have prepared this molecule some time back. It is lying in deep cold storage in Professor Prasanjit's laboratory in the Regional Center for Biotechnology, Faridabad. We do not know. Characterization work has been done by us. We know that this has been formed, but we do not have the data to claim that it is able to do anything. But notice those red spheres, the coronavirus, is being captured by the antenna. That means the hypothesis is sialic acid from both the sides will join and bind and capture the coronavirus cell and kill it. I do not know whether the answer is the yes or no. too early for me to say, because I am not a biologist. I have to wait for a biologist to give me a result, and the biologist has to replicate it 3 times minimum to be sure that his or her results are reproducible. So you cannot make a tall claim that I have done this. But the molecule has been made with the idea that we will capture through that sialic acid in the antenna. All these sialic acids will contribute to the murder or the killing of the coronavirus. That remains a hope. We have not published this work, so I have no right to advertise this work till it goes through the process of refereeing, peer review, and then all the corrections suggested, whatever further data is needed, especially on the biology part, will have to be provided. But as some of you know, this computational work is very difficult because these are very large molecules. To feed them into a computer software is very demanding. So you may get some results if you have improved softwares and predict the properties of such nano sugar balls. One such nano sugar ball has been made by us, but I do not know whether it will be useful. But remember, this is not a biologically produced synthetic vaccine. This is a chemist method working in the industry to produce, such large molecules which have a shape like the immunoglobulin and which should potentially capture the virus or the COVID virus or coronavirus and destroy it. But computational work could be attempted, but computational work with such large molecules is not easy, is quite demanding. Are we through with our lecture, young man? Well, these are patents and publications. We need not look at them. So are we through with, we don't need to look at them. We have seen them on the last occasion as well. So we close today at, I think, less than an hour on a topic which is not difficult. But most of it we had not heard before. We knew glucose, fructose, sucrose, lactose, starch, cellulose. But how many of us knew sugars like sialic acid, glucose, and the others? These carbohydrates or sugars, which are on the outer surface of cells, are very much liked by viruses who attach to this unit and then slowly start doing other things, other bindings to hemagglutinin, and then they cut themselves off from the sialic acid by the time the virus has entered the cell, and then it has infected not only the cell, taken over its machinery, produce more DNA, produce more RNA, produce more viruses, and spread it into the host, and actually kill the host with this infection. But chemists like Caroline Bertozzi and others did a lot of work on these things to understand the mechanism. Understanding the mechanism, they were able to develop chemical drugs which resembled the intermediates or the transition states of these processes. These drugs are available in the international market. We understood that, having understood this, there are sources of glycans being sugar or sugars bound to protein. There has not been much discussion today on the proteins which are there. They are also very important. They form the entire structure called a glycan. But the fact is that these are useful in the treatment of not only viral diseases, but I said for treating a patient like bubble children, or where there are immunocompromised patients in hospitals, and in leukemic cases, these are very useful. Our own very harmless looking pineapple, you can eat the fruit, but you can not just eat the fruit, use the stem also, get its juice, and it contains a glycan called by bromelain. So one of my dreams is, just like this molecule which is on the slide shown, we will have, instead of sialic acid units, we'll have many bromelain units bromelain is pineapple stem juice. It is said if somebody has got a deep cut, it helps the healing process. I did not know this. I did not read about this in my college days or school days, that pineapple stem thrown away, the fruit eaten, the pineapple stem would have such uses. Please look for what is bromelain. On this note, just as we touch the hour, nearly touch the hour, I would say thank you, whoever is present. Very kind of you to listen to me. We are just, I think, a couple of lectures away. The final lecture, I shall give you some interesting stories and anecdotes from another area. On this note, thanking the college, thanking the head of the Department of Chemistry. One of our former students, who is a very senior faculty in IIT Delhi, wrote to me the other day, you are doing a great job. You may not understand everything that I have said today or in the previous lecture, but you must see my commitment. You must see how I am actually working. Under what conditions I worked in St. Stephen's College. Under what conditions I am working today. Most challenging conditions. But when you are most challenged, you come out with your best. And I dare say the period in retirement post 2012, and I think I was told by your head of the department today earlier that one of our students of 2022 batch has contributed brilliantly to a study in Tata Institute of Fundamental Research. I don't know whether Hyderabad or Bombay. I know my student in MRC, Medical Research Council, Tanmay Bharat, around 2008, Rhodes Scholar, RHODES, Rhodes Scholar. He's done such tremendous work. He's got so many awards, publishing mainly in Nature. So chemistry department, students of chemistry department have excelled the recent past or in the previous era. So I congratulate all those who contribute to the success and therefore to the success of St. Stephen's. Let us, our flag of St. Stephen's, fly high and we retain a very challenging position. But please understand, I do not want St. Stephen's to be converted into a research laboratory. I want St. Stephen's to continue to be an excellent place for teaching different subjects. We have very few subjects in college, just a handful. But more important, the fellowship, the relationship between the senior member and the junior member is so intimate. And especially some old students say, if you have not lived on the campus, if you have not been a resident student, then you are not a Stephenian. You are not a Stephenian. You are not a Stephenian. So one must understand that living on the campus, interacting with each other, learning from each other, working together, what makes St. Stephen's College and other colleges great. I'm not talking only of St. Stephen's College. I repeatedly said, now Delhi University boasts of some of the most well-known colleges in the country. And students are coming from all over, all parts of the country, including, I mentioned Hindu College, Miranda House. I did not mention ARSD College, Atma Ram Sanatan Dharma College, Sri Venkateshwara College. They are doing very good biochemistry, biotechnology, and so on. On this note, I stop my lecture for the evening, which is exactly an hour since I started. If there are any thoughts, any comments, any questions, I'm ready to take them. Why should we be discussing the third alphabets of life? Is knowing amino acids and nucleic acids not enough? Yes, it is a regular. Sir, about क्या है सवाल बताओ पढ़ो.
Yes, sir. Yes, sir. Sir, about after an year of pandemic outbreak, there was a disease similar to tuberculosis, which was not diagnosed. What are your insights on that? This is the question from Kirti Rani.
बहुत अच्छा सवाल, बहुत अच्छा सवाल है. क्यों मैं उनको प्रेस नहीं कर रहा हूँ, सच्चाई है ये. Just like you see, 1985, we for the first time heard of HIV/AIDS, autoimmune disease syndrome, right? HIV, AIDS, वायरस है वो भी. और ये पता होना चाहिए कि immune system of AIDS patients is also highly compromised. And one of the problems they always have, they have symptoms like tuberculosis. And most of the tuberculosis already in the world are untreatable because they have developed multi-drug resistance, MDR-TB. So as early as 1985, tuberculosis, Mycobacterium tuberculosis, had become a difficult disease to treat because जितनी दवाइयाँ थी वो चल नहीं रही. Same time, in India, they developed a triple drug therapy. They took 3 different drugs and treated tuberculosis. Then tuberculosis came under somewhat control. But the young person is asking a very pertinent question. What happened was that during the COVID and post-COVID, tuberculosis made a great comeback. And you know, this is a highly infectious disease. Not only COVID-19, but even tuberculosis is highly, infectious, and already it was multi-drug resistant. Near House Cars, there is an institute and a hospital where I went and attended a meeting where many doctors are working on this disease. We had also a project with us from the open source drug discovery of CSIR, where 2 of our molecules, 5,6-dimethoxybenzofuroxane and indoloquinoxylene dioxide, made in St. Stephen's College, we can produce even today kilograms of these compounds, showed activity against Mycobacterium tuberculosis at a concentration of 8 micrograms per milliliter. Whereas the drugs in the market, rifamycin or rifampicin and para-aminosalicylic acid and isonic acid, it's a small molecule, all the 3 are together used for triple drug therapy of patients of tuberculosis, also referred to as Mycobacterium tuberculosis. So the treatment has to be done over a period of minimum 6 months. So if somebody is taking these 3 drugs for 6 months, there are some toxic side effects which are possible. Whereas a low active drug, less active drug may be better in the long run if the patient has to be treated in an appropriate manner. So your question is very good. During COVID-19, it is not just the viral infection, but also tuberculosis made a tremendous comeback, and there were no good drugs to treat patients. And you'll be surprised to know that tuberculosis is not a disease for only the poor people. I know once in the residence of St. Stephen's College, the then president of the Union Society of College, actually one day vomited blood. These people not only throw out phlegm, but also at some stage vomit blood. All that is highly infectious. But they were able to control his disease. He became normal. How can a young man in St. Stephen's College living in the campus, on the campus, having good food, having good surroundings, still get tuberculosis? That means there were some habits, I don't want to pinpoint, which were not helpful to the lungs and the chest. And these habits possibly eventually led to tuberculosis and acute tuberculosis. So COVID-19 was not just the COVID virus, but also complicated by, Mycobacterium tuberculosis, almost multi-drug resistant. That means not treatable by any of the known drugs. So they were searching for new drugs. The open source drug discovery, meaning thereby we will share our knowledge with each other and use it for the treatment of the entire world and the poor world. the demand of paracetamol just to control fever. So great during the COVID, and India was supplying a large amount of it. Not just the, the vaccine, but even simple drugs were being supplied from our country. Your question is a very good one. I do not know whether you liked my answer. You still have a comment on my answer.
सर मैं सुजीत बोल रहा हूँ।
हाँ बोलिए।
मेरे मन में एक क्वेश्चन है, क्योंकि पिछले 2-3 लेक्चर में हमने हमेशा कोई ना कोई नोबेल laureate का या तो वीडियो दिखाया है या उनका कुछ थीम हमने बताया है। ओवरऑल ऑल्दो इंडिया वर्ल्ड में तीसरे रैंक पे आता है सबसे ज्यादा पेपर छापने में। तो मेरा ये क्वेश्चन है, थोड़ा सा हटके है, कि इंडिया अभी तक जो नोबेल प्राइज जीते हैं उनमें काफी साइंस में खास के काफी दूरी हो चुकी है। नए नोबेल प्राइज कब आएंगे? पेपर छपना एक अलग क्वेश्चन है, तो ये कब आएंगे? अगर नहीं आएंगे तो क्यों नहीं आएंगे? आएंगे तो क्यों आएंगे? ये मुझे थोड़ा सा एज ए साइंटिफिक क्यूरियोसिटी।
बहुत अच्छा सवाल है। आजकल हमारे टेनिस प्लेयर्स भी बहुत अच्छा नहीं खेल रहे, पर बैडमिंटन प्लेयर्स और टेबल टेनिस प्लेयर्स और यहाँ तक कि दोबारा वंस अगेन हॉकी प्लेयर्स, क्रिकेट प्लेयर्स को तो पूछो ही मत। दे आर सुपरस्टार्स ऑल ओवर द वर्ल्ड, दे आर dirty rich, but hockey was ignored, table tennis was ignored, badminton was ignored, javelin throw was ignored. They have all people who have succeeded have worked on their own, but they have started very young. मैं समझता हूँ 20-25 साल की उम्र में साइंस शुरू करना बहुत लेट है। कैच देम यंग। देखो सुरेश चंद्र गोपी, प्रकाश पादुकोण, उसकी बेटी को तो सब हम बॉलीवुड से पहचानते हैं, पर प्रकाश पादुकोण को कौन पहचानता है? प्रकाश पादुकोण, हमारे सुरेश गोपी या दिनेश, क्या नाम उनका, गोपी था मेरे ख्याल, वो जो कोच आज बने हैं, वर्ल्ड चैंपियन बने। तो एक बारी उन्होंने जब जीता वर्ल्ड चैंपियनशिप लंदन में, तो प्रकाश पादुकोण को एयरपोर्ट में मिलने के लिए बहुत सारे लोग गए, मालाएं ले गए। They put him garlands. He said, I don't want any garlands from you because I have just won one tournament. Next year I will lose a match or tournament. You will bring me a garland of shoes. He is one who has inspired badminton players. Gopi Chand in Hyderabad has his own academy. So many badminton players have come out. So many table tennis players are now beating Chinese. So many badminton players are beating the Chinese and others from other countries who always dominated at Indonesia. मेरा कहना है हमारा नोबेल प्राइज जीतने का चांस, हाँ, प्रोफेसर CNR राव कहते हैं कई बारी उनका नाम हटा दिया गया। हमारे एक ECG सुदर्शन थे, फिजिसिस्ट, उनको 5 नोबेल प्राइज मिलने चाहिए। कितने? 5 नोबेल प्राइज। ECG सुदर्शन। He became a physicist and a philosopher. He died somewhere, I don't know, one student wrote about his last few days in the Indian Academy of Science journal, magazine. So we have had people, but mostly it appears to be a phenomenon of Indians working abroad. What is the difference between an Indian working abroad, winning Nobel Prizes, be it Hargobind Khurana, be it our own, student of physics turned into a biochemist, studying the structures of riboflavins and what have you, and not on ribosomes in MRC, where our Tanmay Bharat is working now very well. These are all people who have worked abroad. That means they are being given good facilities. मैं बार बार कह रहा हूँ, सेंट स्टीफेंस कॉलेज एक रिसर्च इंस्टीट्यूशन नहीं है। कोई भी कॉलेज दिल्ली यूनिवर्सिटी में कितना ही अच्छा हो, वो रिसर्च इंस्टीट्यूट नहीं बन सकता, बिकॉज ICSIR का बजट 500 करोड़ रुपए। जब मैं हेड ऑफ द डिपार्टमेंट था कई साल, हमारे पास पैसे नहीं होते थे कॉलेज के बच्चों के लैब के एक्सपेरिमेंट चलाने के, और बाजार में कोई आधी बोतल तो बेचता नहीं भाई। 500 ml of this solvent, 500 grams of that compound. So we have to maintain with great care. मैं चाहता हूँ, just like in China, there will be good funding and access to great instrumentation to young people. स्कूल में 8वीं क्लास, 10वीं क्लास में, 12वीं क्लास, कॉलेज में पढ़ाई के साथ साथ मैंने तो ये भी लिखा था मिशन 2050 सेंट स्टीफेंस कॉलेज में, कि ये लिखा था मैंने कि अगला एक चौथा साल हो, 3rd ईयर के बाद 4th ईयर हो, उस 4th ईयर में कॉलेज में ही कुछ गिने चुने बच्चों को रिसर्च करने की सहूलियत दी जाए, मदद दी जाए, तो उनको आखिर ला एक और 4th ईयर की कहीं ढूंढने की जरूरत नहीं। पर वो एक मेरा थॉट विचार ही रहा। तो समरी में कहूँ, यदि हम नोबेल प्राइज जीतना चाहते हैं, जैसे हम बैडमिंटन में अच्छा कर रहे हैं, टेबल टेनिस में अच्छा कर रहे हैं, तो दुनिया भर में से अच्छे अच्छे कोचेस को लाइए, इंडिया के बेस्ट कोचेस को लाइए, बच्चों को उनके साथ बैठने दीजिए। ये हमारी एक सानिया मिर्जा से पहले जो बैडमिंटन में बहुत नाम था, कौन था उसको बताओगे? जो लेडी जो उनकी सुरेश गोपी की, क्या नाम, गोपीचंद की स्टूडेंट थी, अब तो इतना अच्छा नहीं कर रही है, क्या नाम था? सुबह सुबह उसके फादर 20 किलोमीटर 2 व्हीलर में ले जाते और 5:30-6 बजे उसको टेनिस टेबल बैडमिंटन का प्रैक्टिस करा। आजकल जो मैं नाम भूल रहा हूँ, जो आंध्र प्रदेश से बैडमिंटन की प्लेयर है, बहुत नाम कमाया, बहुत पैसा कमाया उसने। इन सबको गुरु, ट्रेनर, कोच मिले। क्या आप सबको कोच मिले हैं? क्या आप 1-to-1 विद CNR राव मिल के बात कर पाए? आप इंडिया के टॉप साइंटिस्ट के साथ बातें कर पाए? उनके साथ उठना बैठना हुआ आपका? हमारे यहाँ कॉलेज में, हर कॉलेज में बहुत अच्छे बच्चे, 96-98% मार्क्स है, फिर भी उनको फीलिंग है कि हम अच्छे, हम उनका प्राइड बढ़ाएं कि आप बहुत अच्छे हैं, आप बहुत काम कर सकते हैं, आप में बहुत काबिलियत है। मैं चाहता हूं कि हमारे टीचर्स जब आखिर में हम ये पूरा सेशन खत्म करें, इस मुद्दे पे कुछ न कुछ कहें कि हमारे बच्चे कितने काबिल हैं। मैं समझता हूं मैं सिर्फ हवा में बात नहीं कर रहा हूं। आई एम सेइंग यस, आवर यूथ आर वेरी टैलेंटेड। One of the previous presidents of United States said, I think it's Obama who said, Indian students don't waste time watching videos. Indian students are very good in mathematics. Indian students are very good in computers. I say Indian students are very good in chemistry. Indian students are very good in physics. Indian students are very good in biology. Remember, Indian students are very good in biology. Even in school, they learn such amount of biology. हमारे पास कोई कमी नहीं है, पर हम कैसे इसको कार्यान्वित करें, कैसे इसको अच्छी अच्छी बातों में, आविष्कारों में, डिस्कवरीज में कन्वर्ट करें। How to convert this into achievement at an international level? I've given a parallel from cricket, badminton, tennis. I've given you other areas where javelin throw or what have you. We have excelled in recent years because coaching facilities are very good. What is क्या कमी है हमारे कॉलेज के बच्चों के पास? और खासकर यदि कॉलेज के रेसिडेंस में रह रहे हैं, तो एक हमारा सेंट स्टीफेंस का स्टूडेंट कानपुर IIT गया। सर, सेंट स्टीफेंस में तो एक आदत है ना, दोपहर को खाना खाने के बाद यदि क्लास नहीं है तो सो जाते थे। सर, IIT कानपुर गया, मेरे को तो शर्म आ गई। As ashamed. Everybody takes up a bicycle and goes to some place to work, do some research work in some lab. That tradition has to be built. Our students who worked for me at midnight or through the day, into the late evening, into the night, they're all professors in America today. तो प्रेरणा मिली, तो हमने रास्ता लिखा। जवाब ठीक है, गलत है, मैं नहीं जानता। सफिशिएंट है क्या जवाब?
जी सर, जी सर, जी, इससे प्रोत्साहित करने वाले शब्द तो हैं, लेकिन हमें लगता है कि और भी बहुत ज़्यादा काम करने की ज़रूरत है। सिर्फ पेपर काम छापने से काम नहीं चलेगा। जनरली मैंने देखा है 4-5 साल से, जब मेरा होशो हवास है रिसर्च में, कि सर इंडिया में तब काम करना शुरू करते हैं जब नोबेल प्राइज मिल जाता है उस कॉन्सेप्ट पे। और वहाँ पे जहाँ मिलता है वो 18-20 साल पहले से उस पे काम कर रहे होते हैं। तो ये इनसाइट्स मुझे लगता है कि कहीं प्रभावित करता है चीज़ों को।
हमारे विपिन अग्रवाल जो CISR में हैदराबाद में हैं, मैंने बताया था, उन्होंने ETH जूरिच स्विट्जरलैंड में Eidgenössische Technische Hochschule ETH में काम किया। वहाँ हर डायरेक्टर नोबेल लॉरेट था। पर आपने ठीक कहा, वो छोटे असिस्टेंट लेक्चरर की पोजीशन से लेक्चरर बन के, रीडर बन के, एसोसिएट प्रोफेसर बन के, प्रोफेसर बन के, डायरेक्टर बन के नोबेल लॉरेट बने। ऐसा नहीं कि नोबेल लॉरेट थे इसलिए उनको डायरेक्टर अपॉइंट किया। उस इंस्टीट्यूट की ग्रोथ उनको नोबेल प्राइज तक ले गई। तो हमारे देयर मस्ट बी ऑर्गेनिक ग्रोथ इन आवर इंस्टीट्यूट्स और स्कूल्स एंड कॉलेज। डोंट इग्नोर our young boys and girls in schools. Encourage them to do science. Shall we now bring our discussion to a close on this note?
Thank you, sir. And thank you everyone for joining on our third day of our lecture series. We will be sharing our feedback form shortly. Please take a moment to provide a valuable insight. And once again, I would like to share my gratitude to Eswaran sir for this informative and engaging lecture today. Thank you.
Thank you so much. I also take leave. Good night.